Okay, so without any further, let me introduce our third keynote speaker. So that's Dr. Benedict Paten. He's a professor in the Department of Biomedical Engineering at the University of California, Santa Cruz, and serves as Associate Director for the Santa Cruz Genomics Institute, kind of a powerhouse institution by all means. He leads the Computational Genomics Laboratory and the Computational Genomics Platforms groups at Santa Cruz, focusing on computational genomics, including the development of algorithms, software, and services to address a variety of biomedical and genomics challenges. Dr. Paten earned his PhD in computational biology in 2007 from the University of Cambridge, and then completed a postdoc with David Hausler at UC Santa Cruz until he started his own research group there in 2011. His research interests encompass genome assembly, genome comparison, human variation, cancer genomics, and many other topics. He has been involved in several large-scale genomics projects, including the Human Cell Atlas platform, the HubMap, GenCode, and the Human Pangenome Reference Consortium. For his innovative work, Benedict is an awardee of the highly prestigious NIH Mirror Award. He's a WMKEC Foundation Grant Award winner. And he's also a contributor to two Guinness Book of World Records. First, for the most complete human genome sequence as part of the TDT consortium, and then one for the fastest DNA sequencing technique for his work developing ultra-rapid sequencing and informatics to aid in the diagnosis of critical care pediatric cases. Relevant to this meeting, Benedict serves as the principal investigator for the AnVIL team at Santa Cruz, where he oversees the development of DocStore, the AnVIL portal, the Data Explorer, and many other components. So welcome, Benedict, to the platform.
Okay, all right, so I'm going to do something a little different today. I'm not going to talk about pangenomes. I'm not going to talk about comparative genomics. I'm not going to talk about spatial or single cell. I'm going to talk about one specific project that we've been running in the lab for the last 2 years or so, that's focused on the application of long reads and specifically nanopore sequencing for rare disease diagnosis, with this question of like, okay, if we've got these long reads, what additional information do they tell us, and how can they help us in this problem of diagnosing undiagnosed patients? And before I start, I want to emphasize this is really work that was led by Shloka, who created many of the slides here. And was a collaboration between Children's National, the Broad Institute, and obviously Santa Cruz, and it's funded by the Chan Zuckerberg Initiative. Okay, so rare diseases. Okay, so broadly they're defined slightly differently in different areas, but roughly there are things that affect people at that affect less than 1 in 1,000 people individually. Okay, but collectively they're a lot more common than you might think. About 1 in 10 people have a rare disease, and that means that hundreds of millions of people worldwide are affected. And in fact, actually, if you add up all the people with a rare disease diagnosis, it actually dwarfs all the people who are diagnosed with, say, cancer and AIDS globally combined. And there are, as I say, they're individually rare, but there are collectively lots of them. There are 8,000 to 10,000 different rare diseases. And what I like about this pie chart is obviously there's a lot of different categories, a lot of different things that can happen. But about half of them, so 45% in this pie chart, are of a neurological basis, right? Often affecting children, often causing different forms of neurodevelopmental issues. So rare diseases, 1 in 10 people, and 80% have a genetic origin, okay? Right? And most of those affect children. The other 20% are things that involve autoimmune disorders. These are things that can happen sporadically, rare cancers that are not necessarily genetic, right? Sometimes they have a genetic component, but there's there's an element of chance there. So putting those aside, I'm going to be focusing on this 80%. And of course, rare diseases are caused by genetics, and many of them are Mendelian or monogenic. So they're caused by mutations in a single gene. Okay, and because they're caused by mutations in a single gene, they follow Mendelian inheritance patterns. So obviously, we have dominant de novo mutations. So we have a dominant mode of inheritance most of the time for these phenotypes, which are often very devastating. They're caused by de novo mutations. Okay, so there are things that arose early on, back in the parents' germ cells or back early in development that cause the condition. And then, of course, there's also a very large category of recessive variants that are either, rarely we have homozygous, so 2 copies of the same mutation that just got inherited. That tends to really happen when we have consanguinity, when we have identity by descent. That's sort of bad luck, where the patient gets, or the individual gets 2 copies. We also have, of course, more commonly compound heterozygotes, which is where you have 2 disabling mutations. So obviously you've got 2 copies, the stuff you got from mom, the stuff you got from dad. And when you have 2 copies, both copies get disabled, then you're out of luck. So broadly speaking, 8,000 to 10,000 rare diseases, and about 7,000 are known to be Mendelian. So actually the large majority of these inherited rare diseases are caused by a single gene. But actually 40%, which is kind of crazy to me, that it's 2024, right? And we've had the human genome for 25 years, but 40% of Mendelian diseases, we know they're Mendelian because we can look at the inheritance patterns. But 40% of rare diseases still don't have a known genetic cause. We don't know the disease gene. And of course, that's really important, right? Because if you are a patient affected by one of these things, then this affects our ability to make a diagnosis. If we don't know what the underlying genetics are, then of course, because oftentimes we have a constellation of different phenotypes that characterize a rare disease, really hard to say you have specifically this. And of course, if you don't know the disease, if you don't know the gene, then you don't have mechanism, or at least you only have guesses of the mechanism. So it's really important for accurate diagnosis and for mechanism, so we can do research, that we identify the gene. And then of course, optimistically, actually, we're increasingly drug development is about repurposing things. If we can discover the gene, then it may turn out that we at least can start to point towards what might be promising therapeutic directions. So this number, this 40%, has to get less, right? We have to get this to as close to 0 as we can. So why? Why in 2024, with many people's genomes sequenced, are we still stuck with this problem? Well, one plausible reason is that some of the genes and variants are just not discovered because we haven't been able to assay them correctly. Now, there's a bunch of reasons for that, but one of them may be that the kinds of variants that we're looking at, or the region that the gene occurs in, is complex enough, right, that it is just kind of that our current genetic sequencing technologies are not able to reach them. So think about structural variants, think about repeat expansions. These are areas of our genomes that we have for the last 20 years not done that good a job of sequencing, because we've predominantly been relying on low cost, very, very fantastic short reads that are necessarily short. Okay. So if you are a patient starting this process of getting diagnosed, you'll typically go through a fairly extensive series of tests. So you may well have a microarray done. It's still done now. It's really useful for establishing copy number variants, for establishing gross level differences across the genome. Oftentimes we'll do gene panels, which are targeted at specific genes that we know, that we know to be disease genes. If the next step, if the gene panel doesn't work, is to do some kind of exome sequencing, right? And then complementary to the genetic analysis that I'm talking about here, we're now, it is not uncommon to do some form of methylation testing. So there, there's a whole bunch of disorders that involve imprinting. So think Angelman syndrome or Prader-Willi. These are syndromes that often involve the methylation of one or other haplotypes. And so we can do methylation testing to understand which bits of the genome are being, methylated and therefore repressed. And of course, we can do whole genome sequencing, which, for the last, forever has been short read whole genome sequencing. And at the end of that process, we're still in a frustrating situation where it depends on the condition, and there's a bunch of caveats to this statistic. But typically speaking, for a patient coming along with a sort of broad array, a broad constellation of symptoms, we'll have a diagnostic rate of less than 50%, or around about 50%, right? Which obviously is, not really acceptable. Yeah, average time to an actual diagnosis is around 5 years. Undiagnosed patients actually sometimes receive different kinds of symptomatic treatment pre-diagnosis. And of course, if you're don't know what you've got, that's really, it's difficult. You're sort of shooting in the dark. You don't know whether the therapeutic is likely to be accurate. And actually, in that time period, 60% of patients actually get diagnosed or misdiagnosed at least once. So, okay. I think this slide was made by Shloka. I think it's a little optimistic, but the intention here with the project that we started with CZ was to say, well, okay, is there a sort of a single assay that could capture all of these things that would allow us to assay the whole genome, allow us to look at the epigenetic state of the genome, right? And sort of understand all the different kinds of variants that a genome contains, and therefore, shorten the time to diagnosis, right? Reduce that 5-year window to something that's much less, right? And have a more comprehensive picture. And yeah, you stole my thunder here. So actually, I slipped this slide in here. When I say much less time, actually, one of the nice things about ONT sequencing, nanopore sequencing, is that the library prep time is actually really short. And I'm talking about hours here. So separate work here, but just inserting this for a second. Couple of years back, my group and you and Ashley's group that sort of led the work at Stanford did this little trial where we brought in 12 patients that were in an acute care setting. So this is patients who've come into the ICU typically, right, and are in an acute situation. So it's an extremely expensive and very time-critical window of time for a patient. And what we did essentially is literally consent them at the bedside and then take their blood draw away and then load up a PromethION, which is a really multiplexed instrument, 48 flow cells on a PromethION, load the sample across all 48 flow cells, right? And then run it so that we could get a whole genome sequence out in about an hour. It's about an hour and a half, I think, right? To get 6dx whole genome. And then we, with a bunch of folks at Google and NVIDIA and folks in my lab, we worked on a pipeline to essentially go from that sequencing to a set of clinical variants, so clinically annotated variants, in about a couple of hours. And so that in total, we were able, on average, I think, our time from blood draw to clinical person sitting down with patient was a little bit under 8 hours, okay? So if we really wanted to, we could do this. Now, I think there are some good reasons why we're unlikely to have clinicians, clinical geneticists on the clock every second, that we can always reach this 8 hours. But at least in terms of the technology, it is absolutely doable that we could in things like acute care settings where it really does matter. If you've had an infant come in and they're in the NICU, that's an $80,000 a day at Stanford exercise, right? So every hour that you save is obviously very important to that patient's life. It's also economically extremely worthwhile if you can identify something that can then, change the course of that person's treatment. So anyway, this was a separate project, but I'm just saying that it is possible to make sequencing extremely fast. And yeah, as a result, because both you and I grew up watching this show in the UK, it was called Record Breakers. Anyway, in which it was all about breaking records. We'd always wanted to have a Guinness World Record. So anyway, so Ewan, and anyway, we're in this book. So I'm going to get a copy for sure. Just found out. So anyway, yeah, fastest genome diagnosis, which I hope in the book it will also have most complete genome as well. So yeah, and this project is scaling up. So I hope that what happened in that setting will be broadly deployed ultimately. Okay. Anyway, so back to that speed. Let's back to like what can these new long read sequencing technologies do for us versus what we've had for the last 15, 20 years. So obviously, longer reads means that we can cover more of the genome, right? And so we're going to have fewer sequencing gaps, right? We're going to be able to assay more of the genome. And in particular, this builds on the previous idea. There are many areas of our genome, obviously, which are highly repetitive. And with short reads, it can be pretty difficult to tell whether or not, if your observation is shorter than the repeat length or some subpart of the repeat length, it can be hard to tell whether or not you've got 3 copies, 5 copies, 10 copies, and what the exact configuration is. And I'll get to an example of why actually having the haplotypic structure, not just knowing the copy number, but having the haplotypic structure is often really important for diagnosis. Okay. And of course, one of the nice things about long reads is that typically, if you think about the human genome as being peppered with variants between the maternal and paternal contribution about every 1,000 bases or so, because long reads, often 30,000 plus bases long, we'll have lots of observations where essentially we can sort out what is maternal and what is paternal, and so phase the data. And this is really important because if we want to find things like, without necessarily having parental information, we want to find out whether something is in cis or trans for a compound het. It's really important to have this phasing information. Okay. And then finally, of course, so if we, long read technologies are typically single molecule technologies. So we're directly reading there's no amplification process, there's no PCR. We're directly reading the original genomic material. We can actually look at the base modifications as they come through the instrument, right? And so detect things like, 5-methylcytosine, 5-hydroxymethylcytosine, and so forth, right? And allow us to sort of distinguish what is methylated and what is unmethylated. And that actually turns out to be really useful. So, okay. So the broad setup of the experiment that we did here was we took a patient cohort. I'll describe that in a little bit of detail. To each patient, we applied high throughput long read sequencing, and as I'll get to, relatively inexpensive long read sequencing. We assembled a workflow and analyzed it, and then, and this was all new for my lab because really we're not clinical geneticists. We developed some, clinical annotation pipelines to try and weed out what was important and what was not. So, in terms of that computational pipeline, so a couple of years back, and this is separate work, my group had got involved with the Centers for Alzheimer's and Related Dementias, which is a new intramural program at NIH that is doing kind of sequence all the things across many different neurodegenerative cohorts. And what they needed from us was a low-cost pipeline that they could apply to essentially do long-read sequencing of a cohort of, I think it's about 4,000 brain samples. And I'm not going to talk about this in more detail, but actually the first papers from that group are just about to come out, and we're well on the way to more than, we're closing in on 1,000 genomes that have been done this way so far. Anyway, the point is that we had a similar sequencing that was being applied, and we needed a pipeline. And so my sort of super postdoc, Misha Kolmogorov, who's now faculty at NCI, developed this pipeline that essentially runs on AnVIL. And in fact, all of the card work is running on AnVIL, right? So it's using Terra, it's using DocStore, all right? It's all written in WDL. And what it does essentially is I won't go through all the details of this, but it takes, our sequencing data, right? It does assembly. So we're doing de novo assembly in this process. We also do reference-based mapping. We do variant calling with, deep neural network variant callers. We ascertain structural variants. We also look at methylation. And when we're done, again, without sort of torturing all the details here, we have a harmonized set of phased small variants and structural variants that are also phased. So anyway, so we took this pipeline that we developed for this brain cohort and said, well, this pretty much fits what we need for this rare disease project. And so, yeah, the output, just in pictorial form, small variants, structural variants, assemblies, which are really the same thing. So, traditionally in variant analysis, we just treat the genome as just a bag of variants. And what I like about this viewing everything as an actual de novo assembly is it allows us to see the context, the actual phasing relationships, and the actual underlying haplotypes. And that's actually a really important and useful view when you're looking at a loci for understanding what's going on. So although this is really just the same thing as this with phasing, it's still a useful way of thinking about the data. And then, of course, we have methylation information. So yeah, key things to think about, and this was all demonstrated in this paper. Actually, SNP performance, just using standard genome in a bottle benchmarks, actually outperforms Illumina at this point. And the reason for that, even though the error rate of the reads is a little higher, is that these reads map everywhere, right, or nearly everywhere. And so we have an appreciable reduction in the amount of false negatives, things that we miss. We miss fewer things. Okay. The structural variances I'll get to are vastly better. And this whole thing was optimized to run, actually it's a lot less than $100 now, but it runs in less than 1 day. I think it's just a few hours now on AnVIL. And as I say, it's really nice because we can run this, and I'll get to some stuff at the end about reproducible compute, but it's really nice that we can run this just so routinely without having to kind of think too much. Okay. So, all right, so back to our cohort. So we had 42 affected individuals, a whole bunch of parents that we also sequenced. A bunch of these, so obviously if we've got the affected individual and the parents, so we call these trios. We also had one, I think, quad, so where we had 2 affected siblings, so same condition but unknown genetic cause, and then 13 cases where we had singletons. 6 cases, so a bit of a weird cohort, but 6 cases we actually picked. These were cases from Children's National where they had already diagnosed this patient with a very specialized clinical test, not short read sequencing, but a very specialized test. And they wanted to see, could long read sequencing, we knew the gene was hard, could long read sequencing basically address them? And then we had 36 cases out of our 42 where these individuals had been through whole genome short read workup. They'd gone through that whole set of assays and they were still undiagnosed. Okay. So in terms of the sequencing protocol, so obviously we have 98 samples total. We have this, I won't run through all the details here, but we essentially, this is work that was kind of led through Karen Meager's lab, but we focused on developing an efficient, low-cost, single flow cell protocol for nanopore. So, wearing my human pangenome hat, in that project, money is an issue, but it's not the driving issue for each assembly, right? We are able to spend a lot of money to generate nearly perfect assemblies, right? And that's fantastic. Our assemblies are $20,000 or something like that at this point. That's not practical for patient care at this point, right? We can't spend $20,000 to sequence an individual. So we really wanted to focus on long read sequencing, yes, but cheapish, right? $1,000 is not cheap, but cheap-ish long read sequencing. That was practical, fast, and all that. So anyway, so these were all basically blood samples taken through a bunch of steps. And I'll show you some data. So from 1 flow cell for each individual, we average about 36x coverage. The mean read N50, so 50%, well, I won't go through the stat, but yeah, 50% of reads are longer, right? Is about 32 kilobases. And the this is all done with the R10 chemistry. So for people who know about ONT's roadmap is just constantly getting slightly better. And so this was done with the R10, not the latest and absolutely greatest R10, but the R10 of about 10 months ago, giving us about a mean read accuracy of about 99%. So 1 error every 100 or so bases. So pretty good. This mixture of good coverage, good read length, and pretty good read accuracy was our input for our pipeline. So taking that's our protocol. Then we take our pipeline, all running on AnVIL. We get these variants out, and then of course we're going to do a bunch of clinical analysis. So going back to these 4 potential areas of improvement, we wanted to systematically go through, and this is really Shloka's work over the last 6 months, go through and just, obviously we wanted to diagnose some of these patients, but we wanted to ascertain in that process what additional information we were getting, like what additional, from a clinical context, from a clinical perspective, what additional information was this long read sequencing providing? So I'll take you through that. So first off, in terms of, just longer reads and fewer gaps, what do we get? Well, actually, this is really building on work from Mike's group and from the T2T work. But previously, people have shown this, and we wanted to see this from a clinical perspective. Here is GRCH38. Here are the chromosomes of GRCH38, right? And all the little blue ticks are regions where there's 1,000 or more bases. See, this is like way zoomed out. So 1,000 or more bases that were callable. That is, there were reasonable numbers of long reads mapping and no short reads mapping, okay? So you can see that there's a whole bunch of blue. And by the way, this pink stuff is the opposite. And actually, most of those are in error. These are places where, I won't go into detail, but essentially they're mismapped short reads. So long and short of it, there's about nearly 1%, 0.8% increase across the whole genome in the addressable fraction of the genome. And again, this is sort of confirming previous results that people have done, but just using our sequencing that we had generated for this project. What's interesting, I think, and perhaps people kind of missed it who hadn't carefully read Mike's paper, if you do the same analysis, but just with T to T CHM13, instead of getting a paltry 0.8%, you're at nearly 6% improvement. Why is that the case? It's because if you think about what GSCH38 is, it's a great reference, but it was ascertained with relatively short, Sanger reads and so forth, right? Relatively shorter reads. And so a lot of the sequence that is unresolved, the gaps in GSCH38 are the repetitive regions, right? Are the stuff that's difficult. And CHM13 being a complete T2T reference fills that sequence in. And so, once you combine an actually good reference, an actually complete genome, with actually much, orders of magnitude longer reads, you get a really substantial amount of the genome that you can see that you couldn't see before. So for me, that number, that's a lot. That's a lot of uncharted territory. Now, admittedly, a lot of that sequence is really difficult sequence. It's very repetitive, complex sequence. But it's still, that's still a lot of the genome to be missing from contemporary assays. And in terms of, again, our focus here was clinical, in terms of just kind of viewing this from a functional perspective, and again, this is building on work from Mike and others who've shown some of this. We see about 280 genes, these are GenCode genes, protein coding genes, I should say here, that have at least one exon that are covered by long read sequencing and not covered by short read sequencing. And in reverse, there are no exons which are covered by short read sequencing, but not by long for whatever reason, right? That's not a thing, right? And actually there are about 38 genes that are fully covered by long read sequencing and fully uncovered by short read sequencing, and about 100 that are about half, right? And actually, if you go and look at known Mendelian disease risk genes, even just comparing, there are about 5 or 6 on average that are covered by long read sequencing and not by short read sequencing. And this number obviously is an underestimate because we're missing and don't know about a lot of Mendelian disease risk genes at this point. So no doubt that number will continue to grow. And then if we look at it from a sort of functional annotation perspective, we see on average about a 10-fold increase in the number of variants that are functionally annotated and callable only with long reads versus those that are callable only with short reads. The ones that are callable only with short reads, these functionally annotated small variants, these are typically currently small indels. Those small indels are the Achilles heel of the ONT platform right now, because it turns out that it's actually pretty difficult to tell the difference between 6 A's and 7 A's, and the ONT platform screws up those variants at a certain rate. And so there are a number, but it's not a large number, of variants that get missed. That's the one thing that you should bear in mind in this. But it is dwarfed by the number of things that are addressable we just could not map with short reads. And actually, if we just then subset to those that are actually really rare in Nomad, so if you're rare in Nomad, you're rare in the population, you're more likely to be of concern. Again, we see a roughly 5-fold enrichment of functionally annotated variants versus short reads. So that's the small variants that we can annotate and know their function. And if you think about, Anshul's talk yesterday, these are the things that we currently can annotate because we know about genes, but there's tons of variants that we have no idea about what they do. So, this is really a tip of the iceberg thing. And then sort of on the other side of things, structural variants. So structural variants are variants that affect 50 or more bases. These numbers look actually kind of comparable. So in terms of long read sequencing, we are assaying of the order of 26,000 structural variants. And we get, 17,000 structural variants out of short reads. But note the incredibly broad distribution here on these violin pots. When we go and look at structural variants, and this is not a new result, that coming out of short reads, they're terrible. I mean, just absolutely terrible. They are, particularly the deletions, just awful. There is just because it's really difficult. It's a fundamentally very difficult problem to call an event that is larger than the size of your read from that data. They're absolutely full of false positives, whereas the structural variants that we get out of the long reads are beautiful, like just beautiful. We have so many really, really, really clean mobile element changes, and of course the NTRs and things involving, you know, short tandem repeats are just resolved, right? Whereas they are not, or at least not completely resolved with short read sequencing. So this is really a night and day thing. I want to emphasize this to people. Okay, so, all right, so I want to talk about some of the actual clinical results we got. So obviously we get phasing out of this without the need for any parental information. And actually in our set, we were able to diagnose an individual using the phasing information. We observed in every sample, we observed interesting compound heterozygous variants. Many of them are what we call variants of uncertain significance, so we don't know what they do. But in this case, we were able to make a clinical diagnosis. So in this case, in this LHCGR gene, don't ask me to say it out, disambiguate it. It's known to cause this Leydig cell hyperplasia. And in our proband, we had a missense variant in this particular exon, right, on haplotype 1, right? And that variant is known, we can see it in ClinVar, it's pathogenic or likely pathogenic. But actually, on the other haplotype, okay, right, we observed, and this was not evident in the short reads, we observe a very clean 6 kilobase deletion, right, that knocks out a fraction of an exon 9 of the gene. And so these two together are obviously disabling both copies of the gene. So, you know, we've mashed up one with a missense variant, we've mashed up the other with a deletion, and so the gene is both copies are unfunctional. And so, yeah, this is the deletion, the red deletion, and then this is the missense variant. So anyway, so this allowed us, and this for comparison, this is what the deletion, when we knew the deletion was there, looks like with the short reads. And there's two things to think about here. One, we don't have phasing information. So we have no way of knowing if this is one or two things going on. You'd guess maybe one, right? So we don't have phasing information. And clearly it's not easy to see the boundaries of this deletion, right? That would be really difficult to call it. So most of these reads it's not showing, but most of, all of the lighter gray reads here, these are low MapQ reads. These are reads that are mapping from somewhere else in the genome, just confusing everything, right? So yeah, in comparison, the long reads is just night and day in terms of what we can see. Okay, so this is a super nice win for the long read sequencing that really allowed us to make a clinical diagnosis for this patient. Okay, so another case. And this is, you know, getting into repeat structure. Okay, so translocations, you know, gross karyotypic translocations happen at an appreciable rate. And they can definitely be detected with short reads using copy number analysis, but it can be really difficult to pinpoint the precise breakpoints and know exactly which portions of the sequence are affected. So actually in our cohort, we had a couple of individuals who had sex chromosomal translocations, as I'll show you. And we knew they had previously, you know, the short read data was conclusive that they had them, but they didn't know the exact structure. And so wanted to see if long reads could like fully capture. And so there's a whole story here about the way in which we did the variant calling, but I'll just show you sort of the evidence here. So we have two patients. These are patients with XX male syndrome. So these are you know, phenotypically male, but they're XX. And what's happened is that a portion of chromosome Y, the male sex chromosome, has got essentially translocated onto the front of one of the chromosome X, leading to a small partial deletion of part of the par of one of the X chromosomes. And actually, this is a copy number plot you can see, but it's pretty clear. So you've got coverage across this portion of chromosome Y, and then it just drops down to 0, right? And then across the par in the other, this is chromosome Y, this is chromosome X. In chromosome X, you can see that we've got roughly 50% coverage across most of the PAR, and then, or actually all of the PAR, right? And then it switches across to diploid. So we've got like a deletion in one of the X chromosomes and a presumable fusion. So that you could see from the short reads. But what's really nice, and again, I'm not going to go through all the variant calling that we did here, right? But just to show you that we had reads that perfectly split map here. So where you've got, here's a read, it's mapping across a portion of chromosome Y, and then it jumps, and this is the supplementary alignment, to that portion of chromosome X. And what's really interesting about this is that this allowed us, because we had precise breakpoint resolution, to know that the portion of chromosome Y includes the SRY gene. So the SRY gene is critical for sex determination, and we had our copy. It's a multi-copy gene, right? We had the SRY gene, and that's what's causing and allows us to be precise about what exactly is going on at the molecular level in this particular translocation. And then this one is kind of similar. It's a slightly different boundaries, but the same thing is going on. Okay, so this is just a nice example where it's like much simpler, much easier to do this kind of diagnosis from long reads. And obviously with the short reads, you can get a picture of it, but it's fuzzy. Okay, so all right, I'm going to come back and I'm going to talk more about structural variation in a minute, but I want to talk about a little tiny bit about methylation first. So methylation information is this ability to see, haplotype resolved information about the underlying methylation of the individual genome. And so this is a really cool story, a really cool case. This is I should say, this is work from Seth Berger, who is PI, I think now at Irvine, but was at Children's National at the time. And so we had this child with a complex non-specific neurodevelopmental disorder, lifelong undiagnosed. And this patient had previously failed whole genome short read sequencing, but we had seen this variant had been seen in the short reads, but it was what we call a VUS, a variant of uncertain significance. And we see it again in the long reads. So I'm showing you in the long reads, but it's a de novo. So we've got mother and father here. You can see that there's no evidence of the read in mother and father, right? You can see that it's clearly present. It's a deeply intronic variant, okay? It has very weak splice AI prediction. So it's a prediction of whether or not it creates a cassette exon. There's weak evidence from splice AI. And this is classified according to ACMG guidance as a VUS, right? Now, if that variant were causative, then this patient could be diagnosed with this Coffin-Siris syndrome. I know nothing about the syndrome, but it's this syndrome that affects this ARID1B gene, right? What's cool is that there is a known episignature with this gene. So there are episignatures are essentially patterns of methylation where clinical folks have come in and said, okay, this subset of CPGs, if they are methylated or unmethylated, right, are predictive of a particular syndrome. And so here is just the absolute slam dunk evidence on our data. When we went in because we knew this episignature existed, took the set of CPGs that were predicted for the episignature and just plot out their methylation pattern. We see that our patient is way outlier in comparison to all of our other samples, right? Like way outlier. So that information, the fact that they have, they're at this outlier episignature with this, what is otherwise just, of us, right? Allowed us to make a confirmed diagnosis. So again, it's really nice to see one assay allowing us to kind of clean all of this information together to pull out a confirmed diagnosis. And actually, we did do RNA-seq for this individual then afterwards. And so we could go in and demonstrate from the RNA-seq that actually what's happening is that variant, that missense variant, creates a little exon in the middle of the intron. So we get this 138 base pair exon, and that obviously just screws up the frame of the gene and it's not expressed. Okay, yeah, you can see this. So this is the RNA-seq. So we're just seeing the splicing I guess this is kind of sashimi plot. You can see the mother and the father, and you can see this additional exon that's being introduced here. Okay. So yeah, across all of these areas, we saw interesting cases, but there was, and this I think is my big take home from this project. In every single case, many of these undiagnosed cases, we saw interesting looking variants, and we would show them to the clinical folks, and they would just be like, yeah, that's interesting, but it's not an OMIM gene, and we don't know. We don't know. We don't know. It's not, we can't make a diagnosis based on that. We don't have enough information. Okay. So I really came away from this feeling like, wow, we can see more, but we don't know what it means. Okay. And so we're going to need to do a lot of work between now and the future, right, to actually untangle this. And I think the only way to get there is by just doing more and more of the sequencing and then depositing these variants in ClinVar, even though they might start as VUSs, depositing these things in Matchmaker Exchange and just moving forwards, hoping to get to N of 2. Okay. But it is interesting that just in the time that we did this study, some of our patients who were previously undiagnosed were diagnosed. And it's not because we found new information, it's because there was new interpretation that was discovered. So this is a really cool one. So there is a lot of people will know about this, but very recently there was this paper, it was in Nature, because it's so interesting. That's not common for these kinds of papers, but it was in Nature because this gene, this RNU2-4, so this is a small nuclear RNA, right? It turns out that there is this recurrent one base pair insertion. And recurrent, it means it happens over and over again, and it is responsible for about 0.5% of all neurodevelopmental delay, right? Did not know this. Suddenly we find this out. It was completely ignored because, it's easy to see why this, these are de novos. It's the same de novo over and over again. People have been filtering this variant out because it's like, oh, we just see this. It's like noise. It's just noise in our variant calling, right? No, it's not. This is a real thing. And when we went back, this paper came out, we went back and looked at our samples. It turned out that 2 of our samples, these are both samples that we have this variant. So even though nothing changed in terms of the actual genetics, because of that discovery, we were able to diagnose 2 patients. And then similarly, we had another patient where there was this de novo 5' UTR splice variant. And that similarly has just recently, the discovery of the disease gene associated with it allowed us to diagnose a patient, right? So 3 out of our 36 undiagnosed patients, just in the span of these 2 years, we could diagnose, we previously couldn't. So I feel like even though it was kind of like we didn't discover new knowledge there, we as in the sequencing didn't unveil new things, it's really kind of positive that we are kind of making progress as a community in that way. Okay, so then the other catalog of things, obviously, we use HG38 because that's where all the annotations are, so GRCH38, to do most of our analysis. But there are definitely, oops, what's going on here? There are definitely issues with using 38, and I'm going to highlight them in a second, or highlight ways that we can move forwards. But a huge reason why we're unable to kind of understand what's going on with our structural variation is that we just don't have the information right now about what to do with a lot of the structural information the structural variant information that we're getting. But thankfully, projects like, yes, the Human Pangenome, the HGSBC, so the Human Genome Structural Variation Consortium, the work from Danny Miller and Evan Eichler to go back and resequence all of the 1,000 genomes with long reads, and of course, all of us, which a bunch of people here are involved with. These projects are starting to churn out at scale population data sets of long reads. And they're producing these, really high quality things. And obviously I stuck this in because I wanted to credit ColorsDB, but ColorsDB, and also I think there's also going to be Nomad, long read, and so forth. These are going to allow us to build, a they're going to allow us to build repositories of what is common, what is typical in the population, and therefore by subtraction, what is not. And that's a key step that we need to get through. So this is going to be just, I think, hugely important for the curation and interpretation of structural variants. I want to emphasize that population level sequencing of quote unquote clinically normal, whatever that means, right, individuals, is going to allow us to get a much better sense of like what variants are common, what variants might be. We actually saw examples where we saw variants that were interesting, then it turned out that those variants were common in African Americans and the samples were African American, for example. Right? It's really important that we have population level sequencing that is spread across diverse ancestry for the purposes of like being able to solve what is common and from what is rare. And this is my like last vignette. I want to talk about complex variants and how the reference can kind of hold us up in ways that we can kind of move forwards. Okay, so segdupes, right? Segmental duplications comprise about 7% of the genome. Many clinically relevant genes, all of those genes that were addressable only with long reads and not short reads, are in segdupe regions of the genome. Okay, segdupes are, they tend to occur. They're either subtelomeric or pericentromeric. They tend to occur in specific regions of the genome, and they are, they're hotspots for structural variation. And oftentimes, because you've got multiple copies of the same thing, we have this process of gene conversion whereby one copy will come in and essentially copy and paste itself to another copy and vice versa. And that turns out to be really important. So it turns out that obviously short reads and segmental duplications is really hard, right? It's very difficult to call these sequences correctly. But long reads, even with long reads, it can be pretty challenging to get the representation right. So I'm going to talk about the RCCX module, which is a not particularly difficult segdupe, but nevertheless was very challenging for what we were doing. Because even with long reads, if you've got something that is identical over 50, 60, a megabase of sequence, it can be difficult to know whether the long read goes here or here, okay? So as part of this project, this was work, again, led by my former postdoc, John. It's now faculty back in France. We wanted to look at ways to essentially map long reads to representations of segmental duplications that would allow us to capture things like gene conversions and partial deletions in an interesting way. And the way we did this is kind of interesting, and it's by no means cooked. This is not something that I think is like fully deployable across the whole genome yet, but it led us down an interesting path that I want to just very briefly describe. So what John did was take the existing, actually GRCH38 reference and claps together the RCCX module. So the RCCX has essentially two copies of this. This contains a bunch of different genes, but for our purposes, we care about this CYP21A2, which is a functional gene, and its pseudogene CYP21A1P, right? So we've got these two copies, and we basically collapse them together to create a pangenome representation of this locus, in which, if you imagine a read threading through here, right, we've got sequence that is common between the two loci, and then we have a bunch of identifying SNPs, right? The green stuff is the stuff from the CYP21A2, and the red stuff is the one from the CYP21A1P, okay? And when we map reads to this, the advantage here is that we don't have to worry about whether or not our read goes to this copy or this copy. We're just going to map our read to this structure and allow the variants to kind of like fall out as they come. And okay, so just briefly in terms of the disease genetics, we know that CYP21A2 disruption causes congenital adrenal hyperplasia, CAH, right? And oftentimes the current clinical test for this is a really tortured bunch of different Sanger sequencing. It's not something you can do with short reads. So you have to do a bunch of these like really difficult Sanger reactions to try and get accurate assay across this locus. And oftentimes, because this is an autosomal recessive condition, you have to have two hits. So you're trying to basically disentangle what's happening in two different the two different chromosomes, that of course each one is structurally variable, has had some disruption that is causing loss of function in both copies of the gene. So in this case, for example, we can have a deletion and then maybe a gene conversion where a piece of the pseudogene gets inappropriately copied into the functional gene, pseudogenizing it. Right? This happens. Okay, so just rushing through this real quick. So when we map our reads to this pangenome representation, right, we can actually label all the nodes, those that are from the pseudogene, those that are from the functional copy, right? And then we can thread our reads through here, right? And then use them essentially to reconstruct the underlying haplotype, because each read is again essentially good evidence of a particular haplotype. So here, for example, this read would support going through the red copy, all right, and then into the green copy in a normal way. So you could go through the red copy and then round and then through the green copy. So this would be the normal structure of the locus, right? But you can also have, depending on the way the read threads through the structure, you can have copies that support, for example, fusion of one to the other and so forth. Okay, and copies where it looks like, oh, it's all green, so it's all across the non-pseudogenized copy, and then suddenly we have a pseudogenation. We have some sort of gene conversion event. Okay, and again, I will not drag you through this, but using this kind of strategy, what was kind of like mind-blowing to me is that we were able to, across I think 4 different patients, in every case, these were patients where the precise configuration of their locus was not known, right? In every case, we were able to fully reconstruct their haplotypes, including things like cases where we had both a gene conversion here, where they just have a couple of pathogenic alleles from the pseudogene that have been transferred across, along with a gene fusion event on the other haplotype that's squashed them together, right? So these are occurring, and you need to think creatively about your reference in addition to thinking about your long reads if you want to be able to actually interrogate these structures. Yeah. Okay. Okay, so I'm going to summarize because I'm running out of time. So in total, I didn't go through all of our solves, but in total, we were able to diagnose 11 of our 42 patients. In my view, this is Sloker's word. All right. Long read sequencing, I think, can provide, everything and more in a cost-efficient setting. Okay, there are definitely some challenges, but we definitely think that even at this point, long read sequencing in this context can provide a next best test. And actually, we're not the only people who think this, and I think we're going to see increasing clinical application of this technology over the next few years, right? So even if it is not the sort of first line of defense, people are going to continue to use short read sequencing because it's just so established. We think that this is going to have kind of broad impact pretty quickly. I think my big takeaway, as I've said, is just that the increased diagnostic yield, it's a moving target, right? We set out to establish what is the increased diagnostic yield with long read sequencing, and I come away with it well, it's definitely there, but it will get more over time because we're going to understand more and more of what's going on. Yeah, and I have 1 minute, but I wanted to very, very, very briefly just talk about the underpinnings of this with AnVIL. Okay, so obviously my group, we, along with OICR and collaboration with Broad folks, developed DocStore. And okay, here is the 1.16 release. Here is our search workflows page. Okay, and notably, I think we're just about to pass 4,000 workflows in the system. We're getting very close. But anyway, and one thing I wanted to point out to you is that we are now using AI. So yeah, ChatGPT to actually go and add in documentation on our workflows, right? It's a nice little, this is the sort of places where AI can be useful. We are scanning, we're scanning workflows where people have not added any documentation, they've just deposited it and they've not told us what it does. And then the AI generates a summary, and we're very clearly flagging this is AI, okay? So it's super, super nice. But anyway, so this pipeline, I told you earlier, it was called NAPU. All right, so if I type in NAPU, and I need to say end to end because there's a bunch of, there are some sub workflows in this, you can see here's our NAPU pipeline. right? Click on it. Here's our workflow. And importantly, you can go run it in any one of a bunch of different platforms that support WDL, okay? If you were viewing, if this was a Galaxy workflow, you'd have a slightly different catalog of places you could go run it, but these are the different places that support running this workflow. There's obviously details on the workflow. And very briefly, one of the things that we've just added is also metrics support. So you can hit up metrics. I promise you, this was just like, I did this, a couple of days ago. Hit up the metrics, and it'll show you that so far this workflow has been run 184 times. Actually, I think this is the R10 version of the workflow has been run 184 times, right? And you can see that there were some failures, but we have 156 completions. On average, it took about 2 hours to run the complete workflow on AnVIL, okay? So this kind of information about being able to show people, like, okay, what is, this obviously isn't cost, but it's still pretty useful in terms of just having some, metric about like how difficult this is to run. All right, I think it's really helpful. And what's important also I want to emphasize is that universally for all workflows in DocStore from maybe next month, everything will have a DOI, right? You'll have a DOI for the overall workflow, and then for every version of that workflow, we will also mint you a DOI if you want to cite that very specific version. For me, that's like a key value thing. People should be depositing their scientific analysis, getting the DOI and putting it in their paper. Like the citation chain should include scientific analysis in precise detail, which is, in my view, because we're combining the metadata, the container, and the source code, it's better than just like putting in a GitHub URL, right? It's a much more precise statement. Anyway, so that's where we're going. And then just to say, that's the preprint. It's about to come out in the American Journal of Human Genetics of the stuff I talked about today. Yeah, I want to say thank you to the many participants in the study, and in particular to Shloka, John, and I guess he's not on here, but Mishra as well in the group. Thanks.
Thank you. That was super cool talk. I was wondering if in the XX male, if you looked at the methylation to see if you saw X chromosome inactivation, and if it was there, was it random or skewed?
Yeah, that's a great question. So we did look at a bunch of imprinting things. I actually don't remember for those samples if we went and looked. Shloka would know. I'm guessing we did, and I'm guessing we did, but that's a not very satisfying answer. I can follow up with you. Yeah. But yeah, the information is definitely there. It's super, super clean. Yeah.
Hi, Ben. This was great talk. So you made a very convincing argument about the use of long rings, right, in solving these cases. So you also presented a story where, you work with a Stanford Medical Center to solve some of these difficult diagnoses. So what would it take, right, to go beyond Stanford and your group, right, to bring this in any, kind of suburban clinic where some of these cases show up, right? So what do you think are the important tools and the necessary steps to democratize these pipelines and workflows that you're talking about?
Yeah, I mean, I think that's a great question, and some of it is a little above my pay grade. So, like some of this is, the way that we value medicine. I would say, technically speaking, actually we're pretty close, right? Like one thing I think that's maybe underappreciated particularly about the ONT platform, is that actually the instrument is really relatively cheap if you're doing, let's say that you're a small hospital that's maybe doing 1 or 2 of these a day. You don't have to spend $250,000 on an instrument. That instrument, P2 Solo or whatever, it's like $20,000, right? It's a really which is kind of like nothing for those kind of institutions. So the actual sequencing infrastructure, I think we could be there. Of course, there's all the regulation and all that stuff, which makes everything take time. But actually, I think in terms of like making that cheap enough, that's already there. I think in terms of the compute, I mean, the fact that we were able to kind of commoditize this and make this run on, cloud platform like AnVIL and just have it be push button, I think that's kind of there too. And there's actually 2 of the students involved in that project have gone off and started like a little mini startup to try and push this out. I don't know whether they'll succeed, but they're definitely, they're definitely trialing it with other acute care settings. And I think it's a pretty easy argument to make that in the case that, you have a child, they have no obvious infection, there's no obvious history of anything that happened, there's no preceding event, and they've had suddenly, sudden cardiac arrest. Like those cases are like so likely to be genetic that it really makes sense. Like it's not every case for sure, but it absolutely makes sense that the genomics should be like front and center in that acute care setting. So I don't know. I think this is one of those things where the technical stuff is all there. Of course, it's logistics and things that have to be figured out, but it wouldn't. That's just no. That's well within our grasp. I think what's much more complicated is the regulatory pieces and all of that stuff, right? So and that's the bit that's above my pay grade. So yeah.
So I have a couple questions. So first one, first of all, amazing work. This is going to be transformative, and I've been a huge fan of all the pipelines you've been developing for many years.
And I want to emphasize that like a lot of what we did is riffing and reusing pipelines that have been developed by many others. Yeah.
So I think I'm not going to get all these, but you presented a compound het, an episignature, sex chromosome translocation, de novo SNP. I mean, it just seems like the big challenge these days is like variant interpretation. We're finding all of these things that have never been seen before, and there's this big challenge there. I'm wondering, is there any clues from in your experience, was there any clues from the phenotypes of like, oh, we think this could be a methylation event, or we think this could be a translocation? Is there some way to kind of go backwards from the phenotype back to the types of variants that were there? Or was it purely, let's look at all the variants that could potentially be functional and then look through them one by one to make these assessments?
No, definitely. So, I mean, for example, if you have a patient with CAH, right? That's a very specific phenotype where they have a very strong suspicion of RCCX being involved. And so in those cases, actually, those were amongst our six cases where we had sort of like a known diagnosis, but they were, there are definitely phenotypes where there's a constellation of genes that are likely to be causative, and your success rate is likely to be much higher than 50%. But then it's the like broad neurodevelopmental ones where the symptom constellation can be incredibly variable and complicated because everybody's presentation can be different. Yeah. That's the hard set. And actually those are the ones where we just saw so many interesting looking variants, and yet couldn't do anything about them. So yeah, no, it's absolutely the case that the phenotypic information combined with the genetic information is it's hugely useful. Yeah.
And maybe in the interest of time, just one last question.
Yeah.
So you are a PI of AnVIL, but you're also a power user. So you have our full attention here. If there's anything that you could ask for to make your life better, what would it be?
Yeah, I should have thought about this question because I knew you were going to ask this question. So yeah, what do I think? I think, where am I going? Yeah, so I think that getting as a group that has just been going through a relatively, this is a smallish scale project, 100 samples, and in the past 100 genomes would be a lot, but now it's a pretty mid-scale project, right? The whole process of going through data deposition and sharing and so forth, and making it easy for people to get their, to go through the process of depositing their workspace and their workflows and all of the pieces, right? So that everything is as reproducible as we can. I think we've really got to push on that to make that easy, because there are yes, there are the big projects like, HPRC and all of us and so forth, which are, huge, and they're going to be coordinated, and hopefully they'll do an okay job of getting their data in. But there's a huge tail of like R01 investigators, right, who are running these kinds of projects, which have loads and loads of value. And if we were talking about some things yesterday about specific databases and stuff, if we are clever about how enabling this technology is, if we can make more and more of our science reproducible and shareable, and it's not just the paper, and we can make that deposition process better, that would be a great thing. So I think for me, even though I'm not really involved in this side of the equation at the moment, I think working through and making it easier and easier and easier for people to deposit their workspace and share their workspace, and figuring out long-term costing and all of that stuff, it's a hard nut to crack, but we need to do it.
Thank you so much.